Hello, and welcome to this lecture on vision. Now, this lecture is really intended for people in a psychology department, taking a course in biopsychology, or maybe senses and perception. It's not really the biology, although there is a lot of biology in this lecture, but it's a little bit about how the eye works and how perception works. So let's get started. There's some really wonderful things happening today in the issues of vision. For example, there are companies creating these bionic or artificial retinas. These are able to take visual information, transduce them either at the retina level or are sending signals directly to the brain. We'll talk a little bit more about these in just a bit. So let's go over some just basic anatomy of the eye. Okay, starting over here, light is entering here, and this is the cornea. It's clear, and it's actually made of the same tissue that your sclera, the white parts of your eye, is made of. It's just made in a way that it's transparent. Light travels through the iris, through the pupils that can get larger or smaller depending on the light. The light is accommodated or bent at the cornea quite a bit, but it is bent also at the lens, and the lens is what's accommodating or focusing that light back here onto the retina. It actually flips it upside down, and you use your brain to flip it right side up. So let's go way back to the retina, all the way back to the retina, and you can see that there are different layers of the retina, okay? And the center part, the part you focus on, is called the fovea, and the fovea we'll talk about here in just a moment, but it's really sensitive to minor changes and it's really sensitive to detail. Then you have the periphery of the eye, the outer part of the eye, and this area is more sensitive to light and dark and movement. Here's another view of the retina. Now light is coming through, and the first thing light is hitting when it's going into the retina, this large tissue here, is ganglion cells. But it really doesn't change the ganglion cells and how they are sending signals. These are the axons coming off of the ganglion cells. The axon of ganglion cells make the optic nerve, cranial nerve number 2. But there aren't many, there are actually a few, but there aren't many ganglion cells that respond to light. Then you have these intermediate cells. These don't respond to light either. These are things like bipolar, amacrian, and horizontal cells. The light actually has to travel all the way back and hit these cells here. These are photoreceptor cells. There are two main types of photoreceptor cells I'm going to talk about. There are rods and there are cones. The cones are sensitive to detail and color. There's a lot of cones at the fovea. The rods are in the periphery of the retina, and they're more sensitive to shades of darkness and movement. But we'll get to that a little bit more. But what's interesting is the light has to come all the way through this tissue here, get into the rods and cones, change the way they are firing, their firing rate, a process called transduction. And then these are sending signals, which is being modified a little bit here at the intermediate cells, and finally sending signals to the ganglion cells. And the ganglion cells are sending their long axons out the cranial nerve, that's the cranial nerve, all the way back to the thalamus. Just another view of light coming through, hitting ganglion cells, not affecting them, hitting intermediate cells, not affecting them, and finally hitting rods and cones and affecting them, and then changing them to neural signals which come back up through here. That's kind of an interesting electron microscope image of rods and cones. You can see why they're called rods and cones. The cones look like cones and the rods look like rods. So the rods are involved in what's called scotopic vision, night vision, right? Different degrees of light and darkness. Achromatic, chromatic means color. These don't work with color. These are in the periphery of your vision. And there are a lot of them, about 130 million. So if you look straight ahead, don't look to the side, What's very difficult to do is notice color in your periphery, in the to the far right or far left. Now your eyes are moving around quite a lot and you're using memories to reconstruct those colors. But really most of the color vision, what's called photopic vision, is right there in the middle, right at the focus, right at the fovea. And the cones are responsible for this. This is daytime vision, color vision, foveal vision. And there aren't as many of them, but they're extremely sensitive. And what they signal, the intermediate cells and the ganglion, those are very sensitive to even the slightest action changes in the cones. So this is just a view of what the whole retina would look like in the entire eye. So this is saying the density of different photoreceptor cells. Recall, cones and rods are photoreceptor cells. And you can see right here in the middle, the fovea, the cones peak. Look how many there are, tons of cones. Now there are a few cones in the periphery, but not many. Not many rods in the fovea, but as you get degrees away from the fovea towards the nose or nasal, or towards the temporal or the side of your head, there are more and more rods. So the rods really peak about 20-30% out on either side. You also have a place where the optic nerve leaves your retina. And there's no photoreceptor cells there. We call that a blind spot. That's where the optic disc is. And you can actually, there's a black spot in your vision, but your eyes are moving around a lot and you're using memories and your perception does not see that black spot. Okay, a few things about some problems with vision that take place at the eye. So, for example, you may have heard of cataracts. Cataracts is a problem with the lens. The lens is becoming cloudy. This oftentimes happens as we get older and older adults, but some people are born with cataracts. This is easily fixable through surgery for those that are able to get it. So you have a clear lens and a cloudy lens, and the light just isn't getting back to the retina. Another common problem, also age-related in many cases, is macular degeneration. And this is where the center, the fovea, begins to degenerate a bit as you get older, and the focus of your attention becomes more and more blurry. It's a very common cause of blindness. Retinitis pigmentosa is another disorder that really goes the opposite way. It starts with problems in the periphery, tissue damage of rods in the periphery, and then it gets closer and closer to the fovea. Also a cause of blindness eventually. Remember at the beginning I talked about those artificial retinas and bionic eyes? These are starting to be used with people with these disorders, macular degeneration and retinitis pigmentosa. Now these are very common problems, and imagine people listening to this wear glasses, and maybe wear glasses since you were a kid. There's myopia and hyperopia. Now this has to do with the shape of the eye. Now sometimes an eyeball can be a little long, like in myopia, and the lens just focuses too close, too in here. Doesn't focus onto the retina, but really accommodates and focuses before getting to the retina. So somebody with myopia, also called nearsightedness, gets concave lenses, which bends the light out, allowing it to accommodate on the retina, on the eyeball, which is just a little longer than normal. Hyperopia is just the opposite. The eyeball might be not long enough, and the accommodation or focus is past the retina. So a concave lens will bend it a little bit more, help that bend so that the focus is on the fovea. Hyperopia is farsightedness. So let's talk about transduction of light. This gets a little chemical, and there's a lot of physiology that goes into this as well, so we'll take our time. Transduction really means the conversion of a stimulus that comes from the outside world, that could be touch against your skin, or sound waves, or light, transduced or changed into the language nervous system can handle, which is really all about depolarizing neurons, neurons sending signals. That's what we're good at. So we have to change, we have to translate or transduce light energy into action potentials or neuron signaling. Okay, so remember that a neuron, a typical neuron, has a cell body and dendrites, and then it sends an electrical signal down the axon, eventually releasing neurotransmitters from these terminal ends of the axon. Well, that's what we have to do. We have to go from light energy to changing the rate at which we get signals down the axon. So let's look at this just in a rod. It's very similar in a cone, but let's look at it in a rod because it just makes it easier to understand, I think. So in a rod, here's a rod, and a rod is kind of a really funny or modified neuron. And it has all these membranes in it, in this sort of modified dendrite. And in the membrane, you have a chemical that responds to light. It's a photopigment. It changes its shape when hit by light. There's a lot of chemicals that do that. Plants have plenty of them because they're using chlorophyll to take in light and change chemicals to make sugars and release oxygen. But we're not plants, but we still need to convert light into something. We're converting light into a signal that we can use. And we're going to use a photopigment in our discussion called rhodopsin. And rhodopsin is made of two molecules, retinol and opsin. So here's my drawing of a membrane in rod. So you have this ion channel here, and it's allowing sodium to constantly come in. It's constantly being kept open. It's kept open by a chemical called cyclic GMP. And sodium comes in constantly depolarizing the cell. This is happening constantly in the dark. Okay? And we call this sodium that comes in the dark current. It keeps this modified neuron, this rod, in a perpetual state of depolarization. And what we have to do is we simply have to change the membrane potential here. We have to change the depolarization. And we do that when light is hitting this rhodopsin molecule. A photon of light comes down, modifies the retinol rhodopsin molecule. It's a process called bleaching because it actually causes them to change color. And this sets in motion a messenger. That messenger is called transducin. So let's look at it again. Sodium is constantly coming in the dark current. A photon of light comes in, changes the shape of the rhodopsin molecule. This sends a message over to cyclic GMP, a transducent, which temporarily denatures cyclic GMP, and the sodium channel closes. Now sodium can't come in. And the cell now isn't as depolarized. We've changed the membrane potential there. All because light hit rhodopsin, changed it, sent a messenger, which denatured cyclic GMP temporarily, closed a sodium channel. Remember, cyclic GMP was keeping it open. Now the dark current isn't happening, and we have gone from a photon of light to changing the ion, the sodium ion flow that comes in during the dark current. Okay, here's just another picture of the same thing I just showed you. Okay, here is transducent affecting cyclic GMP and not allowing sodium to come in. Light strikes the rhodopsin, and this is closing the ion channel. That's all it is. That's all we need to do. We have sodium coming in, and now we don't. We have this neuron in perpetual state of depolarization, and now we don't. And now this neuron is not sending as many neurotransmitters into intermediate cells. We're changing a signal. It always comes down to ion channels. When it comes to neurons and sensory systems, auditory, smell, taste, touch, vision. It's all about having a mechanism to change ion channels. So let's summarize this. I think it's a hard concept. So transduction is the conversion of a physical stimulus to a nerve impulse, or a change in nerve impulse. Transduction takes place in all sorts of sensory systems, but we're talking about vision here. In the dark, opsin and retinol combined makes rhodopsin, are bound together making a photopigment, the photopigment of rhodopsin. The rod membrane is depolarized slightly because sodium is constantly coming in, the dark current. In the dark, sodium is constantly coming in. This sodium channel is kept open by cyclic GMP in the dark. In the light, a photon of light changes the shape of retinol, causing rhodopsin to break apart. It's causing it to change shape. This causes the messenger of transduction to denature temporarily cyclic GMP, and the sodium channel now closes because it's not being kept open by cyclic GMP. The rod becomes hyperpolarized, changing a signal. When we're in the dark again, cyclic GMP is allowing sodium to come in because it's opening up sodium channels. The dark current is back. Light, the dark current closes. That's how we are getting from a light signal to a nervous signal. Okay, now rods and cones, they have a lot of similarity. It's just how sensitive the photopigments are in color vision, how sensitive the photopigments are in cones. They have different sensitivities depending on the cones. And what's called the trichromatic theory says that we have 3 types of cones. We have a blue, green, and red cone. These are selectively sensitive to different wavelengths of light. This is showing the different wavelengths of light. So if we looked at a spectrum of how sensitive they are to different light, you can see that the blue cones are most sensitive, absorb mostly in the blues and the violets. The red right here are highest sensitive to 564 nanometers. That's a wavelength of light. The green, the medium ones, the green, are most sensitive to 534, but they still have some sensitivity here. This level of sensitivity is going to tell just how much or how often these molecules, these retinol and opsin molecules, are going to change shape depending on the light they absorb. And of course, rods have pretty broad spectrum here, and they're most sensitive to 498. So the trichromatic theory simply says we have 3 different types of cones that react to 3 different types of wavelengths of light, generally speaking. We bring that all together, voila, we have color vision. Another theory of color is called the opponent process theory. Certainly the trichromatic theory is correct, but there's probably other components that go into our color vision and color perception. And this is saying that there are ganglion cells in the retina and cells in the thalamus that send different signals depending on if they are excited or inhibited. And remember, ganglion cells are getting information from intermediate cells, which are getting information from rods and cones. And if a ganglion cell is sending a fast signal, a lot of signal, very excited, the signal might be red. However, if it slows down or inhibited, it might be green. So we have ganglion cells and probably cells in the thalamus that are R plus G minus. Red, they're sending a signal of red when excited and a signal of green when inhibited. We also have ones that send signals of green when excited, but red when inhibited. And then we have yellow ganglion cells. These are sending signals when excited that yellow is being perceived. But if inhibited, if these ganglion cells are inhibited, we're sending a signal of blue. And likewise, blue when they're excited, yellow when inhibited. And some of the reasons that we know this happens is, well, there's a number of optical illusions that help tease us out. Let's look at one. Stare in the center. Stare right in the middle. This white dot. Stare at it. Keep staring. Don't look away. Don't look away. Don't look away. Keep staring. Keep staring. Just for a moment, did you see the red, white, and blue flag? Because what happened was the yellow got tired. The yellow ganglion cells got tired. And they've been firing so much because there was so much yellow. They were firing so much because there was so much yellow that when I showed something white, they were actually inhibited, which sends a signal of blue. Same with green. You're staring at the green so long, they're getting fatigued. When you then shift to white, for a moment the image is red because these green cells are fatigued, they've slowed down, they're sending a signal of red. We also have black and white. So that's why the black got inhibited and the opponent was white. Let's see if you see it again. Stare at the middle. Ready? See if you see a flash of the American flag. Just for a second. The longer you stare at it, the longer the after the image. Okay, so let's talk about receptive fields. So remember how I said ganglion cells get information from intermediate cells? And intermediate cells get information from photoreceptor cells, rods and cones? Well, we talk about that as being their field. A ganglion cell has a field that it's responsible for. Now, in ganglion cells, these are round. We call them center surround configuration receptive fields. And what I try to demonstrate here is, let's say this one ganglion cell has a field of cells that send it information. And I kind of put those in dots. These little dots might represent different photoreceptor cells in its field. A ganglion cell with a large receptive field is probably in the periphery, and its cells are rods. A ganglion cell with a small receptive field, a tiny receptive field, still has this donut-looking receptive field, but it doesn't, there aren't as many photoreceptor cells in its field. Intermediate cells are taking information from the photoreceptor cells. Intermediate cells, like Macular cells and bipolar cells and horizontal cells, are getting information from photoreceptor cells and modifying it. They are inhibiting cells to the left of them or inhibiting cells to the right of them. We call that lateral inhibition. It's really modifying what information is coming from the photoreceptor cells before they send a signal to the ganglion cells. And we get what's called on-center and off-center receptive fields that go with ganglion cells. So here's just another piece of information. This bipolar cell, it has a receptive field. And a ganglion cell will be made up of many bipolar cells, which were made up of many ganglion cells. But you can see that the horizontal cells are modifying what signals are being sent. The middle signal is modifying the outer part. We get an on-center or off-center receptive field. I think this is kind of a difficult topic, so let's go through a few slides. So here's one. So let's say, for example, a ganglion cell has a receptive field. And this ganglion cell, this one ganglion cell, is a on-center receptive ganglion cell. It has an on-center receptive field. So let's say this ganglion cell, this is its whole receptive field, and there's light that comes right in the middle. Well, then the signal from this neuron fires a lot. What if there's even more light that goes in the middle, but not in the periphery? A dot of light right in the center of its field. Well, then it's going to fire probably the most it can. This is an on-center receptive field ganglion cell. But what if light starts spilling over into the periphery? Well, then that starts inhibiting the signal. If light is in the periphery of this donut-looking thing, it begins inhibiting the signal. If there's a lot of light on the outside and a lot of light on the inside, we really don't have a lot of signal because we're getting on from the middle and off from the outside, and they're counteracting each other. There are also ganglion cells that are just the opposite. If light hits the middle, they don't fire at all, or hardly at all. And if light hits their periphery, but not the middle, they fire a lot. This is called an off-center receptive field for a ganglion cell. Now imagine lots and lots of ganglion cells with overlapping receptive fields sending different signals depending on how their receptive field is being affected. We start highlighting dark and light edges in our visual field. And we begin to start constructing the things that we're seeing. Here's just another example. I'm just giving you this. This is the on-center ganglion cell. It has an on-center receptive field. Light hits the middle. These dots are representing photoreceptor cells in its field. Look at how much it's firing. When light hits the periphery, it doesn't fire at all. All right? So that's just really the first stage in constructing an image, by looking at light and dark and how they are transitioning between light and dark of an image. So let's talk about the visual pathway. So the optic nerve is really a bunch of axons coming from the ganglion cells. And they are traveling back to the thalamus. And some of those tracks are crossing over at the optic chiasm to the other side, and some are staying the same. Some are going contralateral, and some are staying ipsilateral. Just above the optic chiasm is something called the suprachiasmatic nucleus. This is really important for setting your circadian rhythms, your sleep cycles, your wake cycles. It's getting a lot of information of just how much light is out there. It needs to be reset if you, let's say, travel 12 hours on an airplane. So where it's heading back, where these axon fibers are heading back, is to an area called the lateral geniculate nucleus of the thalamus. From now on, I think I'm just going to call it the LGN, because saying lateral geniculate nucleus of the thalamus is going to get tiresome. Some other information is going to the superior colliculus. There's the superior colliculus and the inferior colliculus. The superior colliculus is really having a lot to do with sort of fast reactions. Getting good information about movements in the periphery. This is your survival thing. Is a ball being thrown at your head? Is there a lion over there in the bushes? This is that kind of reaction. We're not going to really focus too much on the superior colliculus in this discussion. And finally, from the LGN, we're going to get information going back to the visual cortex, way back in the occipital lobe on the back of your head. Let's walk through this whole thing. So, here's the visual pathway. Now, let's take this part. You're staring forward, and things over here in the left visual field, they're going to come in right here. This is going to enter the eye from over here. It's going to come in. We're going to pretend it's the red part, and it's going to hit this part right here, the nasal part of your retina. The nasal part of your retina is going to send its axons to the optic chiasm, and then it's going to cross over contralateral to the LGN, the right side of the LGN, and the right visual cortex. The part against the temple, right here, is going to stay ipsilateral, the same. Going to go to the optic chiasm, it's going to stay on the same side. Now, if you look over here, also in this visual field, in the left visual field, it's going to hit the right eye, and it's going to hit the temporal part of the right eye, and it's going to stay ipsilateral. So the visual field on the left side, hitting here and hitting here, is going to be processed. The left visual field is going to be processed by the right LGN and the right visual cortex. The right visual field, hitting here and hitting here, is going to be processed by the left LGN and left visual cortex. So it's not that one eye goes right or goes left, it's really visual fields. Here's another image showing the same thing. You can trace it. Right visual field hits here, nasal crosses over, contralateral, hits here, temporal, stays ipsilateral. Left, nasal, contralateral, left temporal, ipsilateral. Okay, so here we are at the thalamus. This is this big structure here, and a little place here called the LGN, lateral geniculate nucleus of the thalamus, is getting that visual information. The medial geniculate nucleus of the thalamus is getting information from the auditory system, but the LGN is getting it from the visual system. Right there. There are basically 6 layers of the LGN. There's layers 1 and 2, called the magnocellular layer, and then there's the parvocellular layer, layers 3, 4, 5, and 6. This is actually showing one from a monkey. And the magnocellular layers are getting information from the ganglion cells in the periphery, from the scotopic vision, from the ganglion cells that go into the rods. The parvocellular layer is getting a lot of information just primarily from the fovea, for all that detail that's coming from the fovea. And it depends on the layer of whether it's getting it ipsilateral or contralateral. So, layers 2, 3, and 5 are getting information from ipsilateral eye, and layers 1, 4, and 6 from contralateral eyes. Okay? So we can come back here and we can see this. So this is right here getting the magnocellular layer from a contralateral eye, from a contralateral eye, and from a contralateral eye. Now, the LGN and the visual cortex kind of lay out like a map of the retina. So ganglion cells from one area of the eye is going to one area of the LGN, and the ganglion cells right next door are going to cells right next door in the LGN. We call that a map of the eye. Retinotopic. Topic means map. It's a map of the retina. The LGN is basically a map of the retina, as is the visual cortex. So the parvocellular layer of the LGN, layers 3, 4, 5, and 6, are getting information from ganglion cells, and we call them P cells. P cells are ganglion cells that go to the parvocellular area. The axons from P cells in the retina go to layers 3, 4, 5, and 6 of the parvocellular layer of the LGN. It's easy to remember. P cells go to P, parvocellular. Magnocellular layer of the LGN get information from M cells. Not too hard. M cells are ganglion cells. M cells are not cells in the magnocellular layer. They are in the retina, and they send their long axons through the optic nerve and end up in layers 1 and 2. P cell ganglion cells are smaller. M-type ganglion cells are much larger, and M-type ganglion cells have much larger receptive fields. So if a little bit of light, a little bit of movement happens upon the ganglion cell's receptive field, it reacts to it. That's why the periphery is so sensitive to minor changes in light and minor changes in movement. The P-type ganglion cell has a much smaller receptive field, but it's much more likely to react to even the slightest change here, so it can look and perceive or help in the perception of detail. P cells go to the parvocellular layer, and M cells go to the magnocellular layer. Okay, let's get back to the visual cortex. The primary visual cortex is way back here. Sometimes it's called the striate cortex. It actually has a bunch of names. It kind of drives me a little crazy. So let's look at this really quickly here. Here's the optic chiasm. Here's it crossing over to the LGN. You can see it coming to the LGN. And then these large fibers going back to the primary visual cortex here. Here's another view of it. Now, the visual cortex is oftentimes given numbers: V1, V2, V3, V4, V5. And the information is really going to many of these layers, but primarily starting off in V1. We call that the primary visual cortex, another name for the primary visual cortex, the striate cortex. And we'll see that these different areas take on slightly different responsibilities in the building up of a perceived image. So again, the striate cortex, primary visual cortex, neurons are specialized to respond to aspects of the stimulus such as orientation, movement, size, and direction. These cells are getting information from the LGN. The LGN is getting information from the retina. The LGN is a map of the retina. The primary visual cortex, the striate cortex, is a map of the retina, retinotopic. So recording from single cells in the striate cortex and then presenting visual stimuli to an animal, you can start seeing how they are selectively sensitive to specific stimuli on the place that they are representing in the retina. And this work, Nobel laureate work, was conducted by Hubel and Wiesel, and they did a lot of great work understanding the selective sensitivity of cells in the primary visual cortex of cats. They also worked on the how retina cells, like ganglion cells, had receptive fields and other things like that. You should really know their names. I mean, they won a Nobel Prize. So if we are looking at cells and we are back in the primary visual cortex, these are what are called simple cells. Information from the LGN is coming to these simple cells. And these simple cells are selectively responding to lines of specific orientation on their receptive field. That's in the retina. So let's take a look at this cell right here. If this cell has a receptive field, and if on that field a line that is perfectly vertical happens to be on its field, it fires the most. And if a line on its receptive field is perfectly horizontal, it fires very little. Well, let's look at its next door neighbor. Here's a cell, and this cell might have the same receptive field as the cell next to it. But you know what it likes? If I can say a cell likes something, it likes a line that is not perfectly vertical, but just slightly off. It fires the most. And the cell next to it, it likes a line that is just even off by a greater degree. So let's think of a line, and let's say all of these cells have the same receptive field. And right at that moment, the line is perfectly vertical. This cell fires the most, the next most, the next least, and this one's hardly firing at all. And let's say on that receptive field that the line shifts. And now it is closer to horizontal. Now this cell doesn't fire very much, but this cell fires a lot. You can see how they are selective to specific lines of specific orientation. It's the first stage in building up an image. Here's another kind of pretty picture showing those kinds of things. I'm not going to really talk much about blobs, other than I like the word blobs, but these have a lot to do with sort of color perception. You can see that the left and right eye are both represented in the same area because some information from the left eye ends up here and some information from the right eye ends up here. And these are the lines, these are the cells that like lines of specific orientation. These are called simple cells. And these simple cells will be sending signals to what are called complex cells. Complex cells are getting information from simple cells. They also are responsible for a map. And like simple cells, they like lines of specific orientation. But they like lines that are moving in a specific direction because their receptive fields are larger. They react to movement of lines in a specific orientation, moving in a specific direction. Have we added a little bit of complexity to the building up of the image we're looking at? A little bit. We've highlighted dark and light edges at the retina. We have cells that are firing because of lines of different orientation, the receptive field. We have now cells that are reacting to lines of that are moving in a specific direction, that are of a specific orientation. So here's a complex cell. This is something that Hubel and Wiesel might have recorded. They're recording from a cell. This light, this light of this orientation moves in this direction. It fires a whole bunch. This neuron's firing a whole bunch. Now, it doesn't have quite the same. It's still moving. They're similar, still firing a little bit, but not as much. Maybe if it fires it's completely horizontal, it's firing this way, it's not firing at all. Again, it's all about firing rates of neurons. These lines are building up that image, building up. They might be going also to cells called hypercomplex cells that like lines of specific orientations that have of specific angles. Sometimes these are called stop-end cells. Okay, so now the information that's really started at the ganglion cells, started at the ganglion cells, work their way to the magnocellular layer and the parvocellular layer, are now initiating two kind of pathways of information. We call this the dorsal and the ventral pathway, dorsal ventral stream. The dorsal is really about where is it. It's about location in space of where this image is. The ventral stream, the information coming this way into the temporal lobe, is more about what is it. Is that a square? Is it a circle? Is it a face? Is it a tree? Dorsal is a lot about movement. So let's look at the dorsal stream. This is the where and the how system of perception. So the magnocellular layer, remember, they like movement. It's going to V1, primary visual cortex, V2, V3, V5. This is going this way. V3, V4, V5, this way. That's where the information is being sent. And this is about movement. And people who have damage, perhaps, to this area or to this stream, or specifically to V5, may have trouble perceiving movement. This is a disorder, a visual disorder called akinestopia. Kines means move. A means inability to see movement or perceive motion. And this is kind of a rare condition, but it happens. People see cars moving on a street as snapshots. A car is there, then a car moves and it's there, but they don't see the movement. It's kind of dangerous. The ventral stream is about what things are. This is starting in V1, V2, V4, and going into the temporal lobe, or the inferior temporal lobe. And this is all about identifying and perceiving objects, but it's also about color. V4 is big in color. And we have a condition called cerebral achromatopsia, inability to perceive color. Now, this isn't just color blindness. Somebody with this rare condition can't remember color if they've had like a damage due to a stroke or some other kind of injury. They can't remember or perceive color. And again, it's a rare thing. So let's go down into the inferior temporal lobe down in here. Inferior temporal lobe. And there's an area of the inferior temporal lobe that has a lot to do with objects of vision. So there's one called the fusiform gyrus, this area called the fusiform gyrus, FG, fusiform gyrus. Has a lot to do with images of shapes and faces. Okay, so let's go down into the inferior temporal lobe. These are cells that have receptive fields. They have receptive fields just like all the other cells in the visual cortex. And if on their receptive field they are getting information, enough information about shapes and images and faces, that's when they're responding to the most. They're getting information from simple cells, complex cells, hypercomplex cells. There are cells in the inferior temporal lobe called primary cells. They respond to simple shapes like squares, spots, and circles. You might have a cell that likes spots. It sees spots, it fires the most of spots. It has squares on its receptive field, it doesn't respond a lot. That's its next door neighbor. It's responding to squares and circles. Elaborate cells respond to even more complex shapes and textures and shapes with specific colors. They're tuning, neural tuning into those shapes. in the inferior temporal lobe are also what are called face-selective cells. These are cells that respond to faces. In fact, there's been some research, and they found cells that they call the Jennifer Aniston cell. Jennifer Aniston is a TV star, and they found cells when they were recording from people who were getting surgery because of, I believe, epilepsy or a seizure disorder. These cells were, people are awake during brain surgery oftentimes. They were shown pictures of faces of celebrities. And this area, these cells only responded to faces of specific celebrities like Jennifer Aniston. These cells, these face-selective cells, well, they're probably responding to faces. We're very selective to faces. They've also been shown to respond to other things that people really like. Like if you are a collector of cars, these might respond to a beautiful car. Damage to these cells can produce a condition called prospagnosia. Prospagnosia, inability to perceive faces or recognize people. This can be severe where you don't even recognize your own mother's face or father's or best friend's face. But this is a spectrum disorder oftentimes, and some people are just really bad at faces. I think I have a minor case of this because I don't recognize people I really should recognize. Well, okay, so here's kind of a way of thinking about these temporal lobe these cells. This cell right here fires the most when there's a square, or there's a triangle, or here's an elaborate cell with an oval that's colored in a specific way. They're all working together. Face cells, hand cells, all those kinds of things. They're all bringing in that information and building up that image. This is called a linear processing. At the retina, we're highlighting edges and lines. At the primary visual cortex, at the simple cells, lines of specific orientation. Then complex cells of lines that are moving or have angles. These are hypercomplex cells. We're building them up, and finally we get to these temporal lobe cells that really have the memory of very specific objects. Here are some face cells from research, or face selective cells. They're recording from a macaque here. And when the macaque was seeing a face that's facing them, it fired the most. Fired a little less when we started taking out features, and fired even less when it was a hand. Then there were cells that were firing because of faces of specific orientation. This cell fired the most with a face pointing directly at them, but not in a profile. Another cell fired the most when a face was in profile. We're really sensitive to seeing faces. How many of you have looked up to clouds and seen faces, or seen faces in rocks? You know, some artists, this is Bev Doolittle's work, where I think it's called like The Forest Have Eyes or something. And here it's just a forest with rocks and streams, and all of a sudden you start seeing the faces coming out. They start having enough information. Here's another nose and an eye. How many faces do you see here? You know, the longer you look at this, the more faces you'll see, because we tune ourselves to faces. And this is where we get into the idea of parallel processing, where we actually start getting information from the temporal lobe, going back to the primary visual cortex and reshaping in a way the things we see. It's getting rid of things we don't want to see. It's using memories. This is what you think you ought to see. What is out there and what you see can be very different because you're using memories and context and shading and time of day and all these things to shape what you see, to alter the perception. So here's just a few examples. So there's no white triangle on this page. There's no white triangle. There's these little black, what look like little Pac-Man, right? There's these black things, but there's no white triangle. But you see a white triangle because there's enough information. And in fact, even though there are no lines here or here, when you're just staring at this, you start creating a line because your brain is saying there's enough information that there's a white triangle. I am going to go ahead and perceive a line that's not there. Here's another one I love this illusion. So A and B squares are the exact same shade in every way, the same darkness. And you're like, no, that's not right. B is much lighter of a shade than A. Well, it's because your brain is saying, well, B is in shade. It's being shaded here. And so I must lighten it up to accommodate the shade. So here's a circle. Here's a dark gray circle. I'm going to pass it through. I'm not going to change it at all. I'm just going to pass it through the shadow and tell me if it doesn't change colors. Here we go. Wait a minute. That's the exact same circle? Yeah, I didn't change it. If I remove it, there it is. It's the same thing. Let's do that one again because it's so much fun. Okay? I'm not changing it, but your perception of it is different. Even though what's happening on your retina and your primary visual cortex is all the same. Your temporal lobe is saying, yeah, I know that's what you think you see, but let me help you out. It's much lighter than you think. As soon as I take away context, it's a dark circle again. We do the same thing with color. These two circles are the same color in every way. You say, no, that's green, that's yellow, that's green, that's yellow. But it's about context. If I take this yellow circle and I move it over into the light, you begin to perceive it as green. It's not, it's yellow. But I change it. I move it over, and your brain says, I'm taking into account context, and I'm going to change its perception. You can't do it. You can't help but do this. You can't see it as that. I took away the background, and you can see they are the same exact shade. These are just fun examples about how your brain is really modifying the things that you are sensing in the process of perception. Here's another one. These two shapes are exactly the same. You go, no, this one's much wider. This one's much longer. This shape and this shape are not the same. But you're taking into account context. You're taking into account that this one's going off into the distance and this is going off into the distance. You're taking that into account. Let's highlight it. There it is. That's the shape. I'm going to move it over. I didn't change it. I just moved it into a different context. Same shape. So let's summarize. We get the transduction of light information into neural signals at the retina. Photoreceptor cells are now sending information to intermediate cells and ganglion cells. These are modified. Light edges are modified with center surround fields in the intermediate cells and the ganglion cells. Signals are going out the ganglion axons, P cells from the fovea, M cell ganglion cells from the periphery to the LGN. All the way back to the primary visual cortex where we begin to build the image up. We get a serial processing, also sometimes called linear processing, where we begin to build up that image from lines of specific orientation all the way up to shapes. But then parallel processing, where we're kind of modifying and going back and forth and modifying what you think you ought to be seeing, taking into account memory, context, experience. Context and memory play a big role in perception. I showed you some visual illusions that help demonstrate that, but we're doing it all the time. It's a long lecture, but I hope it was clear.